In this screencast, I'll show you how to start viewing a genome in IGB, load a dataset, load some sequence data, and move around the genome by zooming, scrolling, and double-clicking. When you first launch IGB, you'll see a window that looks like this. Notice this blank region. This is the main data viewer in IGB. This is where data will appear once we've selected a genome and loaded a dataset. The lower part of the display shows a set of tabbed panels that contain various IGB controls and alternative views. I'll use the Data Access panel to control the types of data that IGB will show. Now I'll show you how to load a genome. First, I'll select a species from the Species menu. As you can see, there are a large number of species with sequenced genomes available. To start, I'll choose Arabidopsis thaliana. Next, I'll choose the latest version of the Arabidopsis genome, which was released in June 2009. Notice that if I hold the mouse over a genome option, tooltips with genome version synonyms appear. For example, TER9 is another name for the June 2009 Arabidopsis genome release. When I choose the genome version, a list of data sources that have annotations and other data for that genome appears on the left side of the Data Access panel. I'll choose the BioViz Quick Load data source and check the box labeled TER9 All. When I click the checkbox, IGB adds that dataset to the list of active datasets in the Load Mode table in the middle of the Data Access panel. Now I choose the method of loading I want for this dataset. Because the dataset is small, it only includes around 40,000 gene annotations, I'll tell IGB to load the entire dataset all at once by selecting the Whole Genome option in the Load Mode table. When I choose the Whole Genome option, IGB accesses the BioViz Quick Load data source and downloads the entire dataset into the display. Notice that annotations on the plus strand appear above the number line, and annotations on the minus strand appear below it. The number line represents the base pair positions for the current chromosome, which is chromosome 1 in this case. Now the genome annotations are loaded. To zoom in one of the gene models, I just double-click it. Or I operate the horizontal slider. Notice how during the zooming process the item I clicked stays in view. This happens because IGB allows me to focus zooming by clicking the display. IGB shows me the position of my last click by displaying a zoom stripe graphic, which is the gray vertical line. When I zoom all the way in, IGB displays a gray bar with hyphen characters where the sequence data will appear once I've loaded it. IGB gives me two options for loading sequence data. The Load Sequence in View button will tell IGB to load just those bases covering the region currently in the display. The Load All Sequence option tells IGB to load all the sequence data for the current chromosome, which is chromosome 1 in this case. I'll click Load Sequence in View, which tells IGB to load the sequence data. After it loads, I can zoom in and view the sequence. When I zoom out, the gray bar under the axis tells me where I've already loaded sequence data.